This is our mind map regarding the exocrine gland, and I will be explaining about the glands and types of glands. Epithelia is divided into two main groups according to their function and structure, which are covering epithelia and pelvic epithelia. Glands arise during fetal life from the covering epithelia by means of proliferation or invasion of epithelial cell into subjacent connective tissue. There are two types of glands, which are exocrine gland and endocrine gland. First, exocrine gland consists of a secretory unit, which is a group of epithelial cells that release secretion into urine, and also a duct, which is a tube lined with epithelial cells that transport secretion from the secretory unit to the surface. The formation of exocrine gland. All epithelia begin as simple epithelia that may later become stratified. The epithelial cell will divide by mitosis and grow into the connective tissue or local proliferation of cells in surface epithelium. Complex glands begin to form as epithelial cells grow into the underlying connective tissue. At step 4, downgrowth of epithelial cord, and lastly, the apoplectic cell persists as the lining of the ducts that carries the secretion of the gland cell to the epithelial surface. This is a picture of the exocrine gland. Endocrine gland. First, the connection to epithelial cell is lost and the glands are ductless. Their secretion is picked up and transported by the bloodstream rather than by a duct system. The formation of endocrine gland from step 1 to 4 is the same as the exocrine gland. However, at step 5, the connecting cells disappear. The gland cells secrete directly into the blood vessels or into the surrounding tissue fluid. This is a picture of endocrine gland. And this is a picture of the formation of exocrine gland and the endocrine gland.
Okay, the next one will be classification. Classification of exocrine gland can be divided into unicellular and multicellular.
Next, unicellular glands, such as single secretory cells, for example, goblet cells that secrete mucin. Mucin plus H2O will form mucus. Multicellular glands, simple multicellular glands, secretory epithelial shafts, for example, surface epithelium of stomach.
The next criteria for classifying exocrine gland is duct arrangement. Duct arrangement can be divided into two. First is simple and the second one is compound. Simple gland is a gland with single unbranched duct. Example is sweat gland. Compound is a gland with branched 3 light duct. Example is exocrine section of pancreas.
Now I will talk more about the shape of secretory units.
Next, we focus on the criteria for shape of secretory unit. It can be divided into tube, which will form the tubular gland, sphere or flask, which will form the alveolar gland, and acina, the combination of tubular and alveolar, which will form the tubulo-alveolar or tubulo-acina gland. Simple glands have their secretory unit in the form of tubule present in intestinal gland, coil tubule which is present in the sweat gland, branch tubule in gastric gland, and acinus or alveolar in sebaceous gland. Compound glands can be in acina form which present in the mammary gland, tubular form in bulboretal gland, and tubulo-acina in salivary gland.
Next, we are going to talk about secretory mechanism.
The criteria for secretory mechanism is divided into three types, which is merocrine gland, holocrine gland, and apocrine gland. Merocrine gland, for example, in pancreas, salivary gland, and merocrine sweat glands. The secretory products will leave the cell by exocytosis without the loss of other cellular material. Synthesis of the product will pack in Golgi apparatus, transport to the plasma membrane in the form of secretory granules. Upon reaching the plasma membrane, the secretory granules will fuse with the plasma membrane, then it will excreted out. In sense of the product will also be discharged. In holocrine gland, for example sebaceous gland, the secretory product is shaped with the whole cell. Stem cell will be divided by mitosis. The cell produced will move to the upper parts. When moving upwards, the cell will increase in size. Secretory product is synthesized in secretory granules. Then the secretory granules will reach the upper parts. Thus the cell will burst and releasing all the contents. In apocrine gland, for example, mammary gland, apocrine sweat gland, and prostate, the secretory product is discharged together with parts of the apical cytoplasm. Apocrine gland will accumulate their secretory product at the apical surface of the secreting cell. Then the portion of the cell will pinches off from the rest of the cell to release secretion. The remaining parts of the cell will repair itself and repeats the process.
Next, we are going to talk about types of secretion. Types of secretion can be divided into three: serous gland, mucous gland, and both. Serous gland will produce water secretion. The secretory unit will be serous cell. Apical cytoplasm of the stated cell will contain zymogen granules. Serous cell will form a spherical mass of cell called acinus. Next would be mucous gland. Mucous gland produce mucus. Cells are either cuboidal or columnar. Apical cytoplasm of mucous cell has secretory granules containing mucin. Mucous cells are organized as tubules. Combination gland will contain both mucous and serous cell. This means they will share the same characteristic with mucous and serous gland. However, combination gland will have serous demilunes. Mucous cell will form tubules. The end will be kept with serous cells. This will form a crescent-shaped structure called serous demilunes.